I'm the other Bridget from the University of Sheffield speaking today about my work looking at the contribution of senescence to dementia. So I've had plenty of people introduce dementia today, so just to very briefly reiterate, the most common cause of dementia is Alzheimer's disease, which is typically classified by aggregated amyloid beta plaques and hyperphosphorylated tau tangles. But closely following in prevalence is the mixed dementia, which includes other pathologies in addition to these classical amyloid beta and tau. And this is particularly true of later life individuals, so people in their 80s and 90s, where we also see quite a significant overlap in pathological burden between those with and without a dementia diagnosis. And a lot of the treatments that have been tried that are kind of based on these classical pathologies of the amyloid beta and tau, they haven't performed quite as well as we might have hoped. And potentially one of the reasons for this is that we haven't really considered many of the modulating factors that drives this very complex picture of diagnosis and progression of dementia, as I said, particularly in this later life group. And this brings me to the topic of my talk today, which is the potential role of senescence. So senescence is a cellular mechanism that's very important in development and tissue repair, but we think it may become detrimental as we age. And we know that senescent cells accumulate throughout our lives. And so for my project, I'm really interested in whether senescence is kind of an independent mechanism of aging, or if it does indeed interact with the dementia pathology and contribute to this disease. So a little bit more information about exactly what is senescence. So senescent cells have a number of key features in common. So we see irreversible cell cycle arrest. We know that senescence is at least partially initiated and maintained by the DNA damage response. We see inhibition of apoptosis, metabolic and mitochondrial changes, morphological changes. And we also see this SASP, which involves the release of pro-inflammatory factors that can actually induce further senescence in neighbouring cells. And then this, combined with the fact that senescent cells are resistant to apoptosis, means that we do see an accumulation of these cells as we age. And we also have identified that many of the key features of senescence have also been kind of highlighted as key features in dementia. So particularly things like the DNA damage has been linked to early stage events in Alzheimer's disease, and other factors such as, the mitochondrial dysfunction, neuroinflammation. And this kind of leads us to consider whether senescence is a kind of major pathological feature that interconnects these different factors. This leads to our hypothesis that senescence in the brain contributes to dementia, potentially through loss of function in neuroinflammatory mechanisms. So our aim for this project was to develop a profile of senescence markers to investigate their expression and then their relationship with dementia in a late life neuropathology cohort. So moving on to that cohort, this has already been introduced by my colleague, but I'm also using tissue from the CFAS cohort, the Cognitive Function and Aging Study. So just to reiterate, this is a population-based longitudinal study, and it has some really great advantages in that individuals are not recruited based on any pre-existing diagnostic or clinical criteria. And that's great because it means that we can really examine the full spectrum of pathology present in the population in an unbiased way. So we have 6 centres around the UK which are designed to account for variations in socioeconomic status. We have individuals recruited at the age of 65 and upwards. Currently have about 550 brains donated from this cohort. There we go. So just to briefly explain the methods that we've used for this project. So we have frontal cortex tissue, which we have stained for our various senescence markers, which I will come onto in a moment. We then use a software called CFAS, which allows us to select regions across all the layers of the frontal cortex, and then we can digitalize the slides and determine whether they're positive or negative for our markers, which gives an overall kind of percentage of cells positive for each of our markers. So I wanted to show you some of the initial staining results in our first Cambridge cohort. This comprises 99 individuals. So with this project, we've looked at a number of different potential senescence markers, and it's important to say that there isn't any one marker that can definitively prove a cell is senescent, and instead we look for a panel of markers that are indicative of this senescence phenotype. So the markers that we've chosen to move forward for this project, so first we have P21 and P16, so these are negative regulators of the cell cycle, and of course cell cycle arrest is one of the key features of senescence. I just wanted to highlight that P21 in this initial cohort was expressed in neurons, as you can see with the black triangles, and also in glia with the black arrows, whereas P16 we found to be glial only. We also have gamma H2AX in the bottom left corner. This is a marker of DNA damage, and H3K9ME3, which is a marker of senescence-associated heterochromatin. And then I wanted to show you briefly then some of our initial investigations into these markers in the Cambridge cohort. So the first thing we did was we wanted to see how well our markers were correlating with one another. And we basically found that with our cell cycle arrest markers, so the P16 and the P21, they correlate with each other quite well. And this is kind of what we would expect, because this indicates to us that if senescence is occurring in neurons, it's accompanied by senescence in glia. And those markers also correlated well with our gamma H2AX DNA damage marker, which further supports this concept of DNA damage as kind of maintaining this senescence state. We also looked at this final marker, the H3K9ME3, but we didn't find it to correlate with the rest of our panel, so we actually moved away from using this marker, as it just didn't appear to fit particularly well. And then moving on to looking at the relationship with some of our markers and Alzheimer's type pathology. So first we looked at P21 and P16 in relation to Braak and Thal staging. So here we found that there was a higher level of glial P21 at lower Braak staging, so potentially a little bit contrary to what we were expecting. We didn't find any other statistically significant relationships between P21 and the Thal staging, or between P16 and gamma H2AX and Braak and Thal staging. So what we can say from this is that it does not appear, in this initial Cambridge cohort at least, that senescence is increasing with increasing burden of Alzheimer's type pathology. And then indeed we also went on to look at the relationship between our markers and dementia status, and we did not find any statistically significant relationships. So it may be that senescence is indeed an independent mechanism of aging. It could also be that senescence is linked to dementia, but indirectly, perhaps via another mechanism such as the neuroinflammation. What we're prioritizing for this project is to increase the power of this study by expanding to 2 additional CFAS cohorts to see if the results are consistent with our initial findings in the Cambridge cohort. So just to conclude, we have identified and characterised a panel of markers that appear to be indicative of senescence. We've found that they correlate well with one another and with DNA damage markers. We found that glial P21 expression is higher in low Braak cases, but otherwise there's no statistically significant relationships found between our markers and the Alzheimer's type pathology or overall dementia status. We're currently expanding this work to additional CFAS cohorts, and also looking at the relationship between senescence and potential markers of neuroinflammation, and also currently I've carried out some microarrays looking at gene expression changes in senescent neurons. And I just wanted to wrap up by highlighting the contribution of Irina, who was my predecessor on this project, to thank all of our collaborators and our neuropathology group at Citran, and to thank all of you for your attention for the last talk of the day.
Thank you very much for that, Bridget.
Lots of fun.
Hi, that's a really interesting study. So I'm interested, have you considered looking at sex differences?
Yes, we have that data. I don't think that they found any variation there, but we have a collaborator here at Newcastle, actually, who's a real statistical wizard, who's very good at kind of understanding the relationships between all of our data. And yeah, I do think that that's one of the many things he's looked at. But.
Any question over there?
Sorry, just a question to how you about focus around MSC, which is normal on the screen, and dementia status. If you were to also maybe look at the neuropsychological performance at the time, might be able to find something. And I also wonder, they're mostly people 65 and over?
They're all 65 and over, yeah.
The senescence markers that you identified are actually part of the more upstream process. If you were to apply the same cohort, middle age, when you might find some features there.
That's such an interesting point, and I've suspected the same when I've started to see these kind of lack of relationships in the older aged individuals. I wonder if that we're not really seeing anything, because if senescence is contributing, perhaps it's a very, very early stage. We know that changes are happening in the brain 20 years before symptoms become apparent, so it may be that the contribution happens there, you're right. To your other point, I would love to have the kind of neuropsychological and data throughout people's lives showing the progression. One of the challenges of the CFAS cohort is that because it's been running for so long and different people have been involved with data collection over time, we don't always have the consistency in what's been collected. We kind of have to work with what we have. But in an ideal world, yeah, that would be perfect.
If you're interested in applying it in middle-aged cohorts, I'll contact you about that.
Yeah, that would be fab.
I was just going to ask about middle-aged cohorts, so there's the answer.
That would probably be, if I had like unlimited money and could do anything, that would be where I would want to take this study, is to look at that earlier stage and kind of see, do we see the changes there? Yeah.
So great, we've got some successful collaboration. So that's the end of that session. I'm going to pass over to Lauren and Callum again.
Thank you.